Well, thanks very much, Al, for that introduction, and for the invitation to be part of this session this morning. I'm already kind of energized by the presentation so far. I think it's absolutely fabulous to kind of see this sort of coming together of different groups, different perspectives on things. So I'm going to focus, my area of interest and my background is critical care and surgery. I'm a trauma surgeon by training, and I'm going to focus on critical care. I realize this is a diverse audience, some of whom are primarily methodologists, some of whom are intensivists. But what I want to really start with is the concept that pandemics and wars have really been inflection points in human history. So over time, we are confronted again and again with a crisis, like a pandemic or like a war, that not only challenges people in the moment, but also serves to change the culture and change the world subsequently. This was the First World War, which one of the factors that ended the First World War was the outbreak of influenza in 1918 and 1919, because there weren't soldiers able to go to the front to kill each other. And that really ushered in the 20th century. That was the inflection point between the 19th and the 20th centuries. Another local pandemic, or epidemic, an important one, was the outbreak of polio that occurred in Copenhagen in 1952. This was the moment that intensive care as a specialty was born, because Henrik Ibsen, who was the director of an ICU in Copenhagen, came up with the idea that perhaps they could save children who had been affected by respiratory polio by performing a tracheostomy and ventilating them by having medical students, residents, trainees stay at the bedside 24 hours a day and bag them over that period of time. Huge effort. It required upwards of 1,000 people to do that, but it fundamentally changed the prognosis for polio, and the mortality rate dropped from about 80% to about 30% for those children. And more importantly, it gave rise to the concept that we in the healthcare system can support life regardless of the reasons it's threatened, when it is acutely disrupted, if there's a possibility that things are going to improve. And so we now have intensive care units as a core element of healthcare systems around the world, including in the most poorly resourced areas, that bring multiple different technologies together, primarily to support physiology during a period when it's being threatened. So ICUs don't so much treat diseases as they support physiology. They've had some important consequences, though, because they've actually generated diseases that didn't exist before, that couldn't exist before, because they only exist in patients who've lived long enough to be alive after something that under normal circumstances would have killed them. And it's really the curse, I think, in intensive care is our trying to understand what these are and how best to manage them. Things like the acute respiratory distress syndrome, things like sepsis, things like the multiple organ dysfunction syndrome, they only exist because the ICU exists to allow patients to develop these. And so we've looked at trying to understand the biology. Biology is enormously complex here. More than 3,700 different genes are altered in their expression. And it turns out that in a mouse, you can manipulate many of these, well over 200 of them, and save the mouse from an experimental inflammatory challenge, but none of these things seem to work in humans. So we have this kind of strange dichotomy in intensive care, some of the most complex biology one could imagine, and some of the least effective treatments that one could imagine. And I think it's underlined a few things, one of which is the need for us to be able to work together at scale to solve a biologic challenge, which I think is bigger than any biologic challenge that we faced in medicine before. But the other is to kind of step back and see just exactly what's going on. What can we do? What can we do in the short term? So here's a prototypical ICU syndrome, the acute respiratory distress syndrome, described in the 1960s within 10 years of the onset of intensive care units. Patients who were being put on ventilators were developing this syndrome where they were hypoxic, they had fluffy infiltrates on their chest x-ray, and so we called it ARDS. It was only with the advent of CT scanning that we began to understand ARDS in a more practical kind of sense, because we could see that in patients who developed this syndrome, rather than this being a diffuse lung problem, there were discrete areas of abnormality. And if you look at this CAT scan on the right side of the slide, what you see is in the areas that are anti-dependent, so not gravity dependent, you see cysts forming in the lungs, and in the areas that are dependent, where the patient is lying down, you see consolidation of the lung. And what this says is that this disease, this syndrome, is at very least in part caused by the way we're managing patients. So all you have to do is reduce the amount of pressure that's being applied to the lung to prevent the lung rupturing and forming these cysts, and to flip the patient over, so that occasionally the areas where gravity is causing secretions to occur is now on the front of the patient. And both of those are two very, very effective methods for treating what is biologically a very complex syndrome. So I think it's fair to say that in the intensive care unit, our benefit has come not because we've been able to identify sexy new therapies that target the host response, but because we've been able to improve supportive care. We are able to minimize the iatrogenic consequences of that care, and we've been able to take some well-known drugs and to use them by repurposing them to treat critically ill patients. So another big inflection point for our specialty came with the SARS outbreak in 2003, the H1N1 influenza pandemic back in 2009, and then of course most recently with the COVID-19 pandemic in 2020. Because the very presence of intensive care units meant that the sickest patients suffering from these diseases were showing up in the ICU. SARS was first identified because patients were being admitted to the ICU with an unrecognized respiratory illness. And so the ICU kind of emerged both as the canary in the coal mine that identified a new disease, but also as the place where those patients were going to be managed. And with that, of course, came a desire, in fact, a need to understand how we manage these patients more effectively. We tried during the H1N1 pandemic to launch a couple of clinical trials. As I'm going to discuss later, critical care is interesting in that we do have international connections and collaborations, which means that we can function at a global scale. But it turned out that this proved impossible because the time required to launch a clinical trial at the onset of a pandemic, to get the case report forms, to develop the protocol, to get REB approval, to figure out what the interventions are going to be, there is an inescapable time requirement for that. And this really nice work by Jalali and Anne in France showed that our success we would have been successful had we had another 2 or 3 weeks, but the reality is you don't, and you have to be ready to go to respond to a pandemic before that pandemic actually happens. Coming out of the 2009 H1N1 pandemic, a group of us through 2 organizations, the International Forum for Acute Care Trialists and the International Severe Acute Respiratory and Emerging Infections Consortium, got together to discuss how could we be better prepared from a research perspective for the next pandemic. The key message that came out of that meeting was that if we're going to be ready for the next pandemic, we actually have to be up and running before that pandemic occurs and before we even know what the problem is going to be. So that's a big challenge, but one of the people at that meeting was Don Barry, who's really the godfather of Bayesian statistics, and he had been working on a model called the platform trial, along with Laura Esserman, who's a surgical oncologist from the United States. They developed a trial called the I-SPY trial. And what the I-SPY trial was a trial for women with breast cancer refractory to available therapies, and they would test multiple different therapies on this research platform to decide which ones had the greatest chance of being beneficial and which ones should move into phase 3 trial. So Don suggested that we look at the platform trial as a potential model for pandemic readiness, and that's what we did over a period of a number of years after that. We designed a trial called REMAP-CAP, stands for Randomized Embedded Multifactorial Adaptive Platform Trial. We selected community-acquired pneumonia because by and large, pandemics look like lower respiratory tract infections. And we set very pragmatic entry criteria. And then we asked, can we actually get this study up and going before the next pandemic need, not knowing what that need was going to be. And so it was a cumbersome, slow, perhaps awkward kind of process, but by the end of 2019, there were almost as many patients enrolled as there were investigators in the trial. It was up over 100 at that time. And then, of course, in 2020, COVID-19 became a global problem. And fortunately, REMAP-CAP was well positioned to respond to that. We had domains going that were going to be relevant to the pandemic, and we were able to very quickly activate other ones because we had the networks and the people involved to do that. And so during the COVID-19 pandemic, REMAP-CAP recruited over 10,000 patients with severe COVID-19. We've published 19 reports so far. A lot of these were in high-impact journals, JAMA, the New England Journal of Medicine. And you can see that what we found is that there are some things that seem to make sense that proved to be harmful, some things that you might have questioned that proved to be beneficial, lots of things that were ineffective. And so in real time, this platform could impact the course of the pandemic. Of course, the poster child for platform trials during the pandemic is the recovery trial, based on work that was ongoing in Oxford in collaboration with China, and building on the efficiencies of the UK NIHR, was able to launch a trial for all hospitalized patients that recruited 50,000 patients and made multiple important observations that really did change the course of the pandemic. So I think we saw 2 platform trials at least, which had a significant impact on how patients did during the pandemic. Paul Glasio published this paper in the Wellcome literature, and it shows, I think, very nicely the impact of research in real time during a pandemic. So you see early on, most patients are receiving hydroxychloroquine. There's a lot of enthusiasm for that. We were actually told in Canada that we couldn't not give it because that would be unethical. It was against the standard of care. Our data showed that it was actually killing patients. It was harming patients. People were reluctant to give steroids to a patient with a viral infection, but indeed, by June, we knew that there was a dramatic effect in the sickest patients of receiving corticosteroids. So this happened for perhaps 20 different interventions that we were able to sort out very quickly. What should you use? What should you avoid in the sickest patients? And according to this report, the net consequence for dexamethasone alone was somewhere in the vicinity of 500,000 to 2 million lives saved, both by showing effective and showing ineffective therapies. But what they recognized is the need to learn from that lesson and to figure out how do we take this potentially very powerful tool and make it work more effectively for the future, for crises, but also for how we provide care to patients on a day-to-day basis. So one of the things I think that's really intriguing about the COVID-19 pandemic was how interconnected the world actually is. This maps out where trials were being performed during the pandemic, and you can see that really the world came together around that. Countries that otherwise would be fighting each other were prepared to share data, to manage patients, and to engage in scientific research. So we are now, I think, at another inflection point as a result of COVID-19 that is going to be particularly critical to the clinical trials enterprise and to science in general. And I want to just take a big step back and sort of talk about how science draws conclusions. We depend entirely on the fact that we anticipate that most things in the universe happen randomly. There's sort of a Brownian motion that means if you put a drop of ink in a water, ultimately the whole water will be pale blue because things will have disseminated. When they don't, when you have a deviation from randomness, you have information, and that is the essence of scientific exploration. The fact that stars and dust in the universe form into galaxies is what allows us to understand fundamental forces at the subatomic level, but also forces such as gravity. The fact that crops grow differently if there's something under the soil is what gives rise to crop marks and what has helped identify ancient civilizations. This is a Roman fort identified by crop marks in the UK. You know, for basic scientists, the fact that when you run a protein on a gel, you get different intensity of bands tells us that something systematically is going on there, and that's how we draw conclusions about causality. So in clinical trials, we use a variant of that. We say everything's happening randomly in a population of patients, except for one thing, and that one thing is going to be a treatment, an intervention we provide to the patient. And when we do that, we can draw the conclusion that any deviation from randomness is a direct result of that one non-random thing that was introduced into the care of those patients. And so we understand that in the form of Kaplan-Meier curves, and you look at that and you say, there's a difference there. It doesn't really matter what the intervention is, but it is the stuff of what we do as clinical trialists. But we're moving beyond the model of the conventional clinical trial. For the longest time, really since the work of Bradford Hill back in the 1940s, we've been doing trials that ask a question: what does this intervention do? Does it help or does it harm patients? And we've been doing this as single trials where we set up the infrastructure for the trial, we ask the question, we figure out what size effect we're likely to see, we figure out how many patients we're going to need, we engage sites, we study it, and we either get an answer we like or we don't like. In critical care, by and large, those answers have been, there's not much difference. And it has generated, I think, a level of pessimism about even the research process itself. Whereas, in fact, what it means is the absence of an effect tells you something, but you need to have more sophisticated models to be able to understand what that is. And the beauty of a platform trial is rather than studying an intervention, you're studying a disease. And therefore, you potentially study multiple interventions. So if this is a viral trial or an anti-infectious trial, you can look at the role of remdesivir. But you can also simultaneously look at the role of corticosteroids. You could look at some immune response modifier. You could look at some process of care, like ventilation, simultaneously on the same research platform. You can do that sequentially, you can do that simultaneously, and you can do it perpetually. You set up a structure that is potentially there for as long as the appetite is there to fund it. So there are many different advantages for research platforms. One of the most important is that you're not building and taking down the research infrastructure every time you undertake a new trial. But one of the things that I think is important is clinicians, by and large, don't ask the question, is this drug going to be effective? They say, I have this patient, what do I need to do to help him or her do better? So we're prepared to incorporate this, maybe a drug, it may be a mode of mechanical ventilation. That's what a platform trial can do, because its focus is the problem of the patient, not the particular intervention one's looking at. You can also look at how these interventions potentially interact with each other. Some very cool things out of a platform trial is that when you have multiple sites, the sites can choose which things map to their particular concerns. Potentially a platform model reduces costs, but I think that's still a hypothesis to be proven. But I think probably the most important thing going forward is that it's a great mechanism to build collaboration. So here are data from the pandemic looking at heparin in patients with COVID-19. So collaboration amongst REMAP-CAP and two other platform trials, ATTACK and ACTIVE in the United States. So very quickly, 3 trials got together and recruited over 3,000 patients by the end of 2020 to ask this question. And interestingly, what you see is a dichotomy, that heparin was effective in patients who weren't that ill, but was harmful in patients who were more seriously ill. It speaks to the need for large-scale collaboration, for consideration across a spectrum of illness, and I think it helps to kind of frame where we need to go in planning the research agenda. In acute care, many of us are kind of moving beyond the concept of critical care, what happens in an ICU, to the concept of acute care. These are patients who have injuries, infections in the community. Most of them don't need to come to hospital, Some of them do. Some of them need to be managed in the ICU. Those who are managed in the ICU still have a host of problems after they've been discharged from the ICU. And so we need to think about a continuum of acute care in building research platforms. In Canada, as we've been thinking coming out of the pandemic about this continuity of acute care, we're looking at how platforms can potentially interact with each other over that entire continuum. So Andrew Pinto has developed a platform trial studying patients in the community, patients with moderate disease or mild disease. Ryan Zarachansky, who led the ATTACK trial, has been focusing on patients on the ward. A number of different interventions, practical, remap cap, Panther are looking at patients in the intensive care unit. And then Patrick Lawler has been studying patients beyond the intensive care unit. And we can actually look at these somehow interdigitating with each other so that they become a research process that is carried out over time, so that one could look at how did what happened in the community impact the long-term outcomes of those patients as they are convalescing from their disease. I think platform trials allow another level of complexity, and this is one that hasn't really been explored yet. But in essence, they're a hybrid of research and quality improvement. Some of the features of platform trials allow rapid uptake of information. If you use response adaptive randomization, patients are actually benefiting as the trial is going forward. But we could actually be thinking of randomization not just at the level of the individual patient, but at the level of the hospital or the institution. And thinking about, in a healthcare system, randomizing elements of the healthcare system so that we're continuously learning what works best and what doesn't. That's obviously not going to work for a new therapy for metastatic breast cancer, but it is very effective for those kinds of therapies that we use all the time that we don't know how they work. This is a trial published last year in the New England Journal of Medicine. Laura Lynn McIntyre from Ottawa in Canada came up with the idea of randomizing hospitals to preferentially stock either normal saline or Ringer's lactate. And when a patient was ordered crystalloid, there was an automatic substitution to whichever crystalloid the hospital was participating in this cluster RCT at the time. She was able to recruit more than 45,000 patients to what was in essence a pilot trial. Showed a 1% improvement in mortality, not statistically significant, for patients that received balanced salt solutions, and did this all for $500,000 Canadian. But you can think about this. Almost every patient who comes into a hospital receives a balanced salt or receives intravenous fluids. If there is a 5% relative difference in survival based on the fluid you give, that's a massive impact for the system itself. And so being able to do this at scale, where we say randomized 300 different sites to really answer the question, is not that expensive. It's not that difficult to manage how we could do it. But the system itself could be using the processes of care to answer the question, how do we give that care better? We're working right now with the World Health Organization on a very ambitious model. It's taking a long time to kind of unfold, but thinking about this in the context of providing respiratory support to patients around the world who are hypoxemic, and particularly focusing on the needs of patients in low and middle income settings. So we're trying to set up a research platform that will incorporate multiple different respiratory support trials that can randomize either at the individual patient level or at the institutional level, where we can actually evaluate the implementation of protocols and new approaches to therapy. And so, as I say, it is complex. We're still working out the nuances of methodology, the challenges of funding, but I think it's a real opportunity for our community to come together to think about fundamentally changing the way we do research. So what you'll kind of get here is, I've been using the word platform trial and trial platforms interchangeably. I think we're seeing an evolution that started with platform trials where we focused on the individual trial, solidarity, recovery, remap cap, Panther. But what we're now recognizing is these are exemplars of trials in a particular area. And these trials could collaborate with each other. They could share data, and they could generate answers much more quickly, because we would be focusing on themes relevant to the particular population that we're studying. How would that work? This is indebted to Patrick Lawler for this slide. You suggested the possibility that you would have research platforms. Some could be infection, so there might be the SNAP trial and REMAP-CAP working on the infection side, respiratory failure, Panther, practical, cardiogenic shock might be there. When you have a patient, if the construct was this is sepsis, then they might go into both the infection and the respiratory failure components of that. You might be looking at an intervention where the intervention potentially had benefit across a number of different disease states. For example, endotoxin. Endotoxemia is common in congestive heart failure. Is it a potential target? Well, let's ask it across these multiple platforms. So you can see that you can kind of potentially integrate these within the context of an ICU, a health system, or even a national health system as we think forward. This is critical in critical care because there's probably no other specialty in medicine that requires as many decisions with as little data as we actually have. These are basic questions about what are we going to do here, and there's not clear evidence that this particular drug is the answer. It's been looked at by Scott Halpern, who's calculated that on rounds in the ICU, we make on average 9 decisions about what we're going to do for a patient. Most of those decisions are just made on the basis of throwing up your arms and say, okay, let's do this. If we moved from random decision-making to randomized decision-making, we now have a trial potential that can answer these questions in real time. And so I think that one can think about acute care as a demonstration project for an intelligent healthcare system that's built on research platforms. Multiple reasons for this: the data-rich environment of the ICU, the high costs, the high morbidity and mortality. But the notion would be that you have the care of one patient informing the care of all patients, that information from the patient can be collected electronically and entered into clinical and even biologic databases if you collect samples. The questions can be asked by clinicians at the bedside and then shaped by clinicians working in research networks, and so that you have an ongoing process of discovery. So, platforms, again, they accommodate differences in perspective, and not everyone has to do the same studies in a healthcare system. This obviously involves some fundamental changes in the way we think about things, from changing the research culture, acknowledging that we don't know the answers, that we do trust science, accepting that research is a core skill that any clinician should have. We have to change research methods. We have to rethink the model of informed consent from one of patient protection to one of justice for everyone who's in the system. We have to rethink things like blinding. It may not be necessary, it may not be practical, and it may not have a big difference. We need to think about outcomes, and we particularly need to think about getting patients engaged in this to establish what the priorities are. For those in academia, a whole host of issues come up with group authorship. How do you incorporate this into promotion? How do you reimburse for time spent? A huge issue in low and middle income countries. And then this is all happening in an environment that's changing with electronic health records, with AI. But I think what we need to be doing is moving from a model where funders fund research to a model where the health system funds research, as any other large industry funds research into how to make itself better. And in critical care, we have the advantage that we are working together at scale as networks. This is looking at collaboration across different research networks in critical care. And the details aren't important, but what you can see is connections being made at international scale between networks. And the people who are doing these are people who are coming out of networks of clinicians who are also investigators. This model has taken over in critical care. Something like 61% of all high-impact publications come from these groups. It's a relatively small number of them. And 85% of the work that is published in high-impact journals is looking at how do you improve the use of what we already have, rather than how do you introduce new interventions. There's just a list of some of the groups that have been particularly active in setting this new research agenda and how they can work together. So where we are in physics in the 1960s, if you were an aspiring physicist, all you needed was a basement lab and an oscilloscope, and you'd sit in front of the oscilloscope and you'd start to draw conclusions about electrons and that. That no longer works. Physicians realized in the 70s, 80s, and 90s that they needed larger scale to generate ideas. And so they built the Large Hadron Collider in France and Switzerland. It's the small circle on this diagram. It would cost, I think, $5 billion to produce, entailed thousands of people. But it discovered the Higgs boson, like a fundamental particle in particle physics. Now they want to study the Higgs boson, and they say we've got to have another collider that's 100 kilometers in diameter, and it's in the process of being designed, built, and funded. And they have changed the culture. So we worry about first and last authorship. Physicists don't. On this paper that was the discovery of the Higgs boson, the first author is Dr. Odd. And the reason he's the first author is he has two A's at the start of his name. And they put everybody in alphabetical order. And on that list, I'm not going to show them all, but the list is over 5,000 people on the masthead of a paper. We don't have to solve that problem for journalists. Let them worry about how we acknowledge collaboration in research. So just finally, I think we had a meeting in Kigali last October that's focused on trying to build collaboration in areas where it doesn't currently exist. Brought together researchers from Africa, from Latin America, from Southeast Asia, but 125 people in all, half of them coming from Africa. There is enormous appetite for enormous curiosity and an appetite for changing things. And I think that coming out of COVID-19, we are in a unique position to leverage that to really fundamentally change the way we do clinical research. Thanks very much for your attention.